Welcome. Today we're diving into the fascinating world of PCR and qPCR techniques. These methods have revolutionized molecular biology, allowing scientists to amplify and analyze DNA with incredible precision. In this video, we'll explore what PCR and qPCR are, how they work, and why they're so important in research and diagnostics. Whether you're a student, a researcher, or just curious about how genetic analysis happens, you'll find this journey both informative and engaging. Let's start by understanding the basics of PCR, the foundation upon which qPCR is built. PCR stands for Polymerase Chain Reaction. It's a technique developed in the 1980s that allows scientists to make millions of copies of a specific DNA segment. This process is essential for many applications, from cloning genes to detecting pathogens. The core idea is simple: take a tiny amount of DNA and amplify it so there's enough to study in detail. PCR has become a standard tool in laboratories worldwide, enabling breakthroughs in genetics, medicine, and forensic science. PCR process involves three main steps: denaturation, annealing, and extension. First, the double-stranded DNA is heated to separate it into two single strands. Next, the temperature is lowered so primers can bind to the target sequences. Finally, a special enzyme called DNA polymerase extends the primers, creating new DNA strands. These steps are repeated in cycles, doubling the amount of DNA each time. After about 30 cycles, you can have over 1 billion copies from just one original molecule. To perform PCR, you need a few key ingredients: the DNA template you want to amplify, two primers that define the region to be copied, DNA polymerase enzyme, nucleotides, the building blocks of DNA, and a buffer solution to maintain the right conditions. Each component plays a crucial role in ensuring the reaction works efficiently. The choice of primers and polymerase can affect the specificity and yield of the PCR product, making optimization an important part of the process. Thermal cycler, also known as a PCR machine, is used to automate the temperature changes needed for PCR. This device rapidly heats and cools the reaction tubes, precisely controlling the timing and temperature of each step. Modern thermal cyclers are programmable and can handle multiple samples at once, making high-throughput PCR possible. The invention of the thermal cycler was a game-changer, allowing PCR to become a routine laboratory technique. PCR is used in a wide range of applications. In medicine, it helps diagnose infectious diseases by detecting pathogen DNA. In forensics, it's used to analyze crime scene evidence. Researchers use PCR to clone genes, study genetic mutations, and even identify ancient DNA from archaeological samples. Its versatility and sensitivity make PCR an indispensable tool across many scientific fields. Despite its power, PCR has some limitations. It can be prone to contamination, leading to false results. The technique also requires prior knowledge of the DNA sequence to design primers. Additionally, standard PCR only tells you if a target sequence is present, not how much is there. These challenges have driven the development of more advanced techniques such as quantitative PCR or qPCR. Quantitative PCR, or qPCR, builds on the foundation of traditional PCR, but adds the ability to measure the amount of DNA in real time. This technique is sometimes called real-time PCR. By using fluorescent dyes or probes, qPCR tracks the amplification process as it happens, providing both qualitative and quantitative data. This advancement has made qPCR a powerful tool for diagnostics, gene expression studies, and more. qPCR, the reaction is monitored in real time using fluorescent markers. As the DNA is amplified, the fluorescence increases, and the machine records this change after each cycle. By analyzing the fluorescence data, scientists can determine how much DNA was present at the start of the reaction. This makes qPCR ideal for applications where knowing the quantity of DNA is crucial, such as measuring viral load or gene expression level. Both PCR and qPCR amplify DNA. The key difference is that qPCR provides quantitative data. Traditional PCR only shows if a target sequence is present, while qPCR tells you how much is there. This distinction is vital in many fields, such as clinical diagnostics, where the amount of a pathogen can influence treatment decisions. Understanding these differences helps researchers choose the right technique for their needs. qPCR relies on fluorescent dyes or probes to detect DNA amplification. Common dyes like SYBR green bind to double-stranded DNA and emit fluorescence when excited by light. Alternatively, probe-based systems use labeled oligonucleotides that emit a signal only when the target DNA is present. These methods allow for highly sensitive and specific detection, making qPCR a reliable choice for quantitative analysis. Data from qPCR is analyzed by plotting fluorescence against cycle number. The point at which the fluorescence rises above the background is called the threshold cycle, or CT value. Lower CT values indicate higher amounts of starting DNA. By comparing CT values to standard curve, scientists can calculate the exact quantity of DNA in the sample. This quantitative power sets qPCR apart from traditional PCR. qPCR can be used for both absolute and relative quantification. Absolute quantification determines the exact number of DNA copies by comparing samples to a known standard. Relative quantification, on the other hand, measures changes in gene expression by comparing the target gene to a reference gene. Both approaches are widely used in research, depending on the experimental goals. qPCR requires specialized instruments equipped with optical systems to detect fluorescence. These machines can process multiple samples simultaneously and provide real-time data analysis. Modern qPCR instruments are user-friendly, with software that guides users through setup, data collection, and interpretation. The integration of automation has made high-throughput qPCR experiments more accessible than ever. Multiplex qPCR allows the simultaneous detection of multiple targets in a single reaction. By using different fluorescent probes, researchers can analyze several genes or pathogens at once, saving time and resources. This capability is especially valuable in clinical diagnostics, where testing for multiple infections or genetic markers in one go can speed up decision-making and improve patient care. Reverse transcription qPCR, or RT-qPCR, is used to measure RNA levels. In this method, RNA is first converted into complementary DNA, cDNA, using reverse transcriptase. The cDNA is then amplified and quantified using qPCR. RT-qPCR is widely used to study gene expression, detect RNA viruses, and monitor cellular responses to treatments. Proper controls are essential in qPCR experiments to ensure reliable results. Negative controls help detect contamination, while positive controls confirm that the reaction works as expected. Reference genes are used for normalization in relative quantification. Including these controls helps identify technical issues and increases confidence in the data. Standard curve is a crucial part of quantitative qPCR. It's generated by amplifying known concentrations of DNA and plotting their CT values. This curve allows researchers to determine the amount of DNA in unknown samples by comparison. Accurate standard curves are essential for precise quantification and reproducible results. Melting curve analysis is often performed after qPCR to verify the specificity of the amplified product. By gradually increasing the temperature and monitoring fluorescence, researchers can detect the melting point of the DNA. A single sharp peak indicates a specific product, while multiple peaks suggest nonspecific amplification or primerdimers. This step adds an extra layer of quality control to qPCR experiment. Even with careful planning, PCR and qPCR can encounter problems. Common issues include no amplification, weak signals, or nonspecific products. Troubleshooting involves checking reagent quality, optimizing primer design, and adjusting cycling conditions. Keeping detailed records and systematically testing variables can help identify and resolve issues efficiently. PCR and qPCR are invaluable in medical diagnostics. They enable rapid detection of infectious agents, genetic mutations, and even cancer markers. During the COVID-19 pandemic, RT-qPCR became the gold standard for detecting the virus. The speed, sensitivity, and specificity of these techniques have transformed how diseases are diagnosed and monitored. In forensic science, PCR is used to analyze DNA from crime scenes, helping to identify suspects or victims. Even tiny amounts of DNA from hair, blood, or saliva can be amplified and matched to individuals. The reliability and sensitivity of PCR have made it a cornerstone of modern forensic investigations. Researchers use PCR and qPCR to study gene function, regulation, and expression. These techniques allow for the cloning of genes, detection of mutations, and analysis of genetic variation. By enabling precise manipulation and measurement of DNA, PCR has opened new frontiers in genetics and biotechnology. PCR and qPCR are also used in environmental science to detect and quantify microorganisms in water, soil, and air samples. These methods help monitor pollution, track the spread of invasive species, and assess ecosystem health. The ability to detect even low levels of DNA makes PCR-based techniques ideal for environmental monitoring. Food safety labs use PCR and qPCR to detect pathogens like Salmonella or E. coli in food products. Rapid and accurate testing helps prevent outbreaks and ensures that food reaching consumers is safe. These techniques are now standard in quality control for the food industry. In agriculture, PCR and qPCR are used to identify desirable traits in plants and animals. By screening for specific genetic markers, breeders can select individuals with the best characteristics, speeding up the development of improved crops and livestock. This molecular approach enhances traditional breeding methods. One of the most important uses of PCR and qPCR is detecting pathogens in clinical, environmental, and food samples. These techniques can identify bacteria, viruses, and fungi with high sensitivity and specificity, often within a few hours. Early detection is critical for controlling outbreaks and protecting public health. qPCR is the gold standard for measuring gene expression levels. By quantifying mRNA, researchers can study how genes respond to different conditions, treatments, or diseases. This information is vital for understanding cellular processes and developing new therapies. PCR-based techniques are used to detect genetic mutations associated with diseases. By designing primers that target specific mutations, scientists can quickly screen samples for genetic disorders or cancer-related changes. Early detection of mutations can guide treatment decisions and improve outcomes. qPCR is widely used to measure viral load in patients with infections like HIV or hepatitis. By quantifying the amount of viral DNA or RNA, doctors can monitor disease progression and response to treatment. Accurate viral load measurements are essential for effective patient management. Maintaining high quality in PCR and qPCR experiments is crucial. Regular calibration of instruments, use of high-purity reagents, and strict contamination control help ensure reliable results. Laboratories often follow standardized protocols and participate in proficiency testing to maintain accuracy. PCR and qPCR are playing a growing role in personalized medicine. By analyzing genetic variations, doctors can tailor treatments to individual patients, improving effectiveness and reducing side effects. This approach is transforming healthcare, making it more precise and patient-centered. Scientists use PCR to study evolutionary relationships by comparing DNA sequences from different species. This helps reconstruct evolutionary trees and understand how organisms are related. PCR has made it possible to analyze ancient DNA from fossils, shedding light on the history of life on Earth. Archaeologists use PCR to analyze DNA from ancient bones, teeth, and plant remains. This allows them to study past populations, migration patterns, and domestication of plants and animals. PCR has opened new avenues for understanding human history and prehistory. In biotechnology, PCR is used to engineer organisms, produce recombinant proteins, and develop new drugs. The ability to amplify and manipulate DNA has accelerated advances in synthetic biology and genetic engineering, leading to innovative solutions in medicine, agriculture, and industry. PCR and qPCR are now standard topics in biology education. Many schools and universities offer hands-on PCR labs, giving students practical experience with these essential techniques. Learning PCR skills prepares students for careers in research, healthcare, and biotechnology. Commercial PCR kits have made the technique accessible to a wide range of users. These kits provide pre-mixed reagents and detailed instructions, simplifying setup and reducing errors. The availability of reliable kits has helped standardize PCR protocols across laboratories. Automation has transformed PCR and qPCR workflows. Robotic systems can prepare samples, load thermal cyclers, and analyze data with minimal human intervention. This increases throughput, reduces errors, and frees up scientists to focus on experimental design and interpretation. Digital PCR is an advanced technique that partitions the PCR reaction into thousands of tiny droplets. Each droplet acts as an individual reaction, allowing for absolute quantification of DNA without the need for standard curves. Digital PCR offers unparalleled sensitivity and precision, especially for rare mutations or low-abundance targets. Working with PCR and qPCR requires attention to safety and contamination control. Wearing gloves, using filtered pipette tips, and maintaining clean workspaces help prevent cross-contamination. Proper disposal of reagents and samples is also important for laboratory safety and environmental protection. Interpreting PCR and qPCR results requires understanding the data and potential sources of error. Analysts look for expected band sizes in gel electrophoresis or correct CT values in qPCR. Careful interpretation ensures that conclusions drawn from the data are accurate and meaningful. While powerful, PCR and qPCR have limitations. Inhibitors in samples can affect results, and primer design remains a challenge. Quantification can be influenced by reaction efficiency and instrument calibration. Being aware of these limitations helps researchers design better experiments and interpret results more accurately. PCR and qPCR technologies continue to evolve. Advances in microfluidics, automation, and data analysis are making these techniques faster, more sensitive, and easier to use. Emerging methods like digital PCR and portable devices are expanding the reach of PCR beyond traditional labs, bringing molecular diagnostics closer to the point of care. Portable PCR and qPCR devices are enabling rapid testing outside of traditional laboratories. These point-of-care systems are used in clinics, field settings, and even at home, providing quick results for infectious diseases and other conditions. The accessibility of PCR is transforming healthcare delivery worldwide. Sum up: PCR and qPCR are foundational techniques in modern biology and medicine. They enable the detection, quantification, and analysis of DNA and RNA with remarkable precision. As technology advances, these methods will become even more powerful and accessible, driving new discoveries and improving lives. Thank you for joining this exploration of PCR and qPCR techniques. Stay curious and keep exploring the wonders of science.